All right, so this is the second audio lecture for Chapter 5, looking at some of the key slides from Chapter 5, which covers microbial growth. Our first audio lecture looked at cell division and population growth. So we're going to be looking at environmental factors and then some additional slides looking at like natural versus laboratory growth and some enumerating cell growth. Now, when we're looking at different influences on growth of microbes, there are a number of different environmental factors that play a role. So thinking back to the first part, we already mentioned about temperature and also the type of media it's in. So most organisms are going to have certain optimal conditions that allow them to grow. And again, that's what we'll focus on for most of this audio lecture. Most organisms like a fairly moderate environmental conditions. And I will say, I feel like that statement from the textbook is a little biased. So we as human beings and our ability to culture microorganisms really are only able to culture about 1% of the microbes on this planet. We know that there are microorganisms that actually thrive in really harsh conditions. So there are the extreme thermophiles or extremophiles. So those are the ones that are going to be found in thermal vents. Those are lava fields. We're good at culturing things that can live on us and grow in us because we clinically want to research them. So yes, most of the microorganisms that we grow live in fairly moderate environmental conditions. Honestly, again, I don't really think it's fair to say that most microorganisms just grow in there. There are microorganisms that grow on ice fields in really exotic climates. And so, again, as we said in Unit 1, there are microbes all over this planet. Every surface has microbes growing on it. And so some of those conditions are really harsh that we wouldn't be able to survive in. The key is if we want to be able to culture them, we need to be able to recreate that environment that they live in and through various factors beyond just temperature, oxygen concentration, pH, salt concentration, water concentration. All those factors need to be controlled for. And again, that's where from a laboratory standpoint, we're not as good at. We're getting better. We're coming up with new techniques that help to mimic some of those different environments beyond just like what we as humans or some other basic organisms provide for microbes. So as I was mentioning, there's a number of factors that do affect the growth, osmotic conditions, and that's focusing on like the water activity. Water activity refers to the water being available for the microbe, and there's a lot of factors that go into that. Within there, your textbook also includes halophiles, halo meaning salt, so these are salt-loving bacteria. And so there are also bacteria that do not do well in salt environments. So again, if we think of high concentration of salt like in the ocean or in the Dead Sea, there are microorganisms that can thrive in that environment. When we look at pH, and this is the first factor that we'll look at in a little bit more detail, there are various pHs that microorganisms can live in, and so we can characterize the microorganism by their ideal pH, just like we can characterize a microorganism by their ideal temperature and their ideal oxygen concentration. So looking at pH, as most of you are probably familiar with at this level, pH is a concentration of hydrogen ions, and so we refer to things as being acidic, neutral, and basic. So acidophiles are going to be able to live in acidic environments where the pH is between 0 and 5.5. So when we think about an acidic pH and thinking about human beings, that might be something like in your stomach, right? Your stomach has a lower pH, so we might see acidophiles being able to populate the stomach, but they're not going to be able to live in other areas of your body which have a more neutral pH, Neutrophiles will like the neutral pH because they grow optimally in pHs 5.5 to 8. And then alkaphiles are the ones that live in a basic pH, so their optimal growth is between pH 8 and 11.5. So most bacteria and protozoas are neutrophiles, which is good because we provide them with that environment, except for in a couple locations. Most fungi actually prefer a more acidic surrounding, so they like a lower pH between 4 and 6. And so if we think about where we might get fungal infections, like our feet, our skin has a more acidic pH, and so sometimes we get fungal infections there. If we think environment, we're more likely to see mushrooms and those kind of like fungi growing in more acidic environment, like where we have pine needles and such found. There's also photosynthetic protozoans. They seem to favor slightly acidic environments. Most of our archaea are acidophiles. And then alkaphiles, we actually see those distributed between the 3 domains of life. Okay, and we just have our pH chart here with the concentration of hydrogen versus our pH, examples of where we see these different pHs. And then you'll see some of these different organisms and what their optimal pHs are. Okay. Now, when we look at temperature, we want to keep in mind that microbes can't regulate their internal temperatures. They are, we're talking about like prokaryotes or single-celled organisms. And so they need to be in an environment that's ideal for their enzymes in order for their enzyme to be functional. So remember, enzymes are proteins. Proteins will denature in really hot environments, and they may not function properly, optimally at lower temperatures. And so these enzymes that these bacteria have are going to have optimal temperatures to function, and that means the bacteria then is going to have an optimal temperature. So on humans, we can regulate our temperature so it is optimal for our enzymes. Microbes can't do that. They have to be in an environment that is their favorable temperature. And that's one of the reasons why there's certain organisms that will affect us as humans, but they're not going to do well in a thermal vent. And the microorganisms that love lava fields are not going to tend to infect us. Okay, so again, each organism is going to have their minimal temperature, maximum temperature, but what they want to be is in their optimal temperature. So here we can see the different groups of microorganisms based on their temperature range. Again, keep in mind when we're looking at each one of these curves, if we are on the lower temperature, that's going to be minimal. The top of the curve is going to be optimal. And then on the right side, the higher temperature is going to be maximum. And so I want you to kind of focus on the green, which is the mesophiles. So the mesophiles, you'll notice that their minimal temperature is 20 degrees Celsius. Their maximum temperature is 45 degrees Celsius. And if you look at their ideal temperature, it's going to be about 37 degrees Celsius. 37 degrees Celsius is the human body temperature. So mesophiles make up a really important group clinically because your microorganisms that are going to grow well in us humans as the host in either symbiotic relationships or pathogenic relationships, parasitic relationships are going to be the mesophiles. Okay, and that is because we are the incubator and we are the ideal temperature for mesophiles. Now, you'll notice that if we just shift our temperature a little bit, we start to move into the less than ideal conditions. We're not optimal anymore. And so that's one of the reasons why you'll get a fever when you have an infection, because you're basically changing the setting on your incubator and you're trying to make it a little less optimal for those microorganisms that are growing in you. So just to link this clinically. Now you'll notice below that we have psychophiles. psychotrophic phials and psychotolerant phials. So these are going to like lower temperatures. So these are the ones that we'd find in like icebergs and ice caps. And there are actually microorganisms that can. There's this one that actually looks pink. And so when like snowbanks have this microbe growing in it, they call it watermelon snow. And I used to tell elementary school kids this about this watermelon snow, and they're like, "Oh, could you eat it, Dr. Schreiner?" And I was like, "Well, you could. Well, would you get sick from watermelon snow?" And I'm like, "Well, no, because like you're not the right temperature, right? Those microbes can grow in the snowbank, but you can't, like, you're not a snowbank, right? Your temperature, your thermostat is set higher, so you're less than ideal. Wouldn't recommend going out and like eating watermelon snow, and that's pink snow, not yellow snow, so don't either of those. You probably shouldn't eat. But again, the kind of comfort here is these are not, you are not the ideal temperature for them. You will then notice there's the thermophiles. So the thermophiles are between 45 degrees Celsius and 85 degrees Celsius. So these are a lot of the soil microbes. So this, thinking about the sun beating down on the soil, that's going to have a hotter temperature than like us, especially around summertime, right? If you have like a compost pile, what a really important step with the compost is letting it heat. So like old timers will be like, the pile's got to get hot. Well, the reason why the pile's got to get hot is so that you're now having thermophiles growing in there, and anything that we might have put in that would be a mesophile that could potentially cause disease is no longer going to be able to live in that compost. So there's really great research papers that have looked at composting toilets and using that material as like fertilizer. NASA actually did this. They used the material after it had been composted and had gone through this heating cycle. As the microbes are metabolically active in breaking down that material, the pile naturally heats. And again, what you're going to see is this population of thermophiles being present. Any mesophiles that were present in that composting from the toilet material, fecal material, would actually die out in that hotter temperature. And then we have the hyperthermophiles. Those are going to like temperatures above 85 degrees Celsius to 100 degrees Celsius. So 100 degrees Celsius, boiling water, right? And so that's wicked hot in my books. And so that would be like the thermal vents, the lava fields. So again, you can appreciate on the planet, there are different areas that have different temperature ranges, and so we can have microbes that live in all these different areas. And then if we think clinically with these mesophiles, the trick is moving outside of their ideal temperature range, making sure they have less than optimal settings in us, the host. Again, keep in mind temperature is one aspect. We already talked about pH. Another aspect we can look at is oxygen concentration. So each microbe is going to have their ideal oxygen concentration, and that depends upon the microbe's metabolic processes, electron transport chain, terminal electron acceptor used. So keep in mind that you do not always need to have oxygen as a terminal electron acceptor. There are other terminal electron acceptors, and we'll talk later about that when we talk about microbes and metabolism. So we have, just like we had different groups for temperature, different groups for pH, we have different groups, terms for different groups of various oxygen concentration. And they kind of break down into two groups. There's aerobic, which utilize oxygen and need to have a higher concentration of oxygen. And then there are anaerobic. Anaerobic can grow in the absence of oxygen. And then there's variations in between that. Okay, so again, aerobic, it is going to grow in the presence of oxygen. And keep in mind, in the atmosphere, the concentration of oxygen is 20%. So that is high amounts of oxygen. So there are some that are obligate aerobic, so they require oxygen to be present. They need it as the terminal electron acceptor. It is not able to metabolize without oxygen being present. We can also have obligate anaerobes, these actually get killed in the presence of oxygen. So oxygen can actually be very damaging. It can create free radicals. And so if you are an aerobic bacteria, you actually have enzymes that allow you to deal with those free radicals so your cells don't get damaged. We as humans have a number of enzymes that allow us to live in a very oxygen-rich environment. There are certain microbes that do not have those enzymes. So the presence of oxygen will kill them because free radicals are created and it will they don't have the ability to deal with that, and that damages their cells and they end up dying. Okay. There are micro aerobophiles, so they require somewhere between 2 and 10% oxygen. And so they're not killed in the presence of oxygen, but they don't need high concentrations of oxygen. So they're not like the obligate aerobics. There's facultative anaerobes. They don't require oxygen, but they grow better in its presence. And there's aerotolerant anaerobes. They grow with or without oxygen, but they grow fine in the absence of it. So the anaerobic, aerotolerant, I think of it as like a take it or leave it. Like, okay, if oxygen's there is fine, but I'm not like, I have some enzymes that can deal with those free radicals, but I'm not going to actually utilize it in my metabolic pathways. All right, so as I mentioned, there's certain enzymes that are present. And so on the bottom of the sphere, you can see SOD. So that is superoxidase dismutase. So superoxidase dismutase, or SOD, is an enzyme that allows the cell to deal with free radicals. There's also catalase. Catalase is another enzyme that allows the cell to deal with oxygen species. So you will end up later on in the semester doing a catalase reaction. So whether a cell, whether a bacteria is catalase positive or negative is one of the definitive tests. It's kind of like the Gram stain, right? If you're wondering what bacteria you have, you do a Gram stain, and you'll probably do a catalase test, and that actually provides you with a lot of information. The reason why we're looking at this is because in the test tubes, you will notice there are the different groups of bacteria based on their oxygen demands. And so the first tube is the obligate aerobic. And so you'll notice how they're growing on the top of there. So the green dots indicate bacteria. So they grow on the top. Towards that top where the media is interacting with the air, that's where the highest concentration of oxygen is going to be in. So they're obligate. They require to have high concentration of oxygen. And that's because they are positive for enzymes that allow them to deal with reactive oxygen species. And you'll notice that there's no growth in that area lower, because that area lower is going to have lower concentrations of oxygen. So we're no longer 20%, we're going 18, 20, 16, 13, 10, 5, right, as we move down that test tube. So we're no longer in that ideal condition. Microaerobophiles, they're positive for SOD. They can be positive or negative for catalase. So they have one enzyme that allows them to deal with the oxygen species. You'll notice that they're not in that high concentration, but they're also not living in the really low concentration. So again, they like to be in this between 2 and 10% concentration of oxygen. Facultative anaerobic, they can deal with oxygen present there, but they're happy without having oxygen. So they're kind of like the aerotolerant anaerobes, but they're a little bit better at dealing with the oxygen, and because they are positive for 2 of these enzymes, SOD and also catalase. Aerotolerant, they lack catalase, but they have SOD. And so again, they're kind of take it or leave it, and so they can be spread throughout. And then a strict or obligate anaerobe, they lack both of these enzymes. And so they're going to find their growth pattern in a test tube is going to be in the lower concentration of oxygen towards the bottom. Okay. So again, you might, in the lab, when you inoculate your bacteria, it doesn't matter where you inoculated it, they will start to exhibit these growth patterns over time. And so that's something to pay attention to, because that can help to identify what organism you have, in addition doing tests like the catalase test to determine whether you have that enzyme present or not. The catalase test, you add hydrogen peroxide, and catalase is able to convert hydrogen peroxide, which is a reactive oxygen species, into oxygen harmless forms of it. So again, as that kind of figure was showing you, there are different enzymes like superoxidase dismutase, SOD. There is catalase. There's peroxidase. These enzymes, as indicated by the ACE at the end, will help to basically deactivate reactive oxygen species, or ROSs. So superoxidase radicals, hydrogen peroxide, hydroxyl radicals, those are all fall into the ROS, reactive oxygen species. So when oxygen is present, when it's utilized, you get the formation of ROSs, so you need to have enzymes in order to deactivate those so they don't damage your cells. An organism that lives in high oxygen concentration, like atmospheric oxygen at 20%, would have these enzymes in order to protect them. All right, so next we're going to be looking at microbial growth in natural environments. So we want to keep in mind microbes in their environment, that their environments can be constantly changing. They're complex. Generally in an environment, it's not that the nutrients are like X concentration. There's actually gradations of different nutrients in different environmental factors. So if we were to look at a soil sample, it's not that the whole entire soil, the whole entire sample, like if we were to take a core sample, it's going to have the same exact nutrients, the same exact temperature, same exact pH throughout the whole entire thing. It's going to have gradation. This allows those microbes to then thrive in the areas that are ideal to them. And we also want to keep in mind that within their environment, there are going to be other microorganisms and even macroorganisms. So again, if we look at our soil sample, we're not going to have just microbes present. We're also going to have insects present, worms, right? And so if we look at us as the environment for a microbe, again, there are going to be other microorganisms present that it's competing against. And there's going to be other macroorganisms present too. So we want to keep in mind that most microbes actually live in a growth arrest state. So when we're looking at that growth curve and when they're in their natural environment, they're kind of at that long-term stationary phase. And that's because most microbes actually inhibit an allogotrophic environment. So there's actually a lack of some of the nutrients they need in order to survive. So they kind of have to wait for there to be nutrients around. So if they're in that environment, they've already gone through the lag phase, they've already gone through the exponential phase, they're in stationary, they've had a death phase, and now they're actually like living long term in that environment. And they might have periods where they don't have enough nutrients. Some microbes do live in a nutrient liable environment where they might be able to utilize certain nutrients that are available and then convert it, right? So we talked about the cyanobacteria, that even if the nitrogen in their environment's unusable, that they have the mechanism undergo nitrogen fixation so they can make usable nitrogen for themselves. But if they're making it in excess, other microorganisms around them can thrive, which then means they're the friend everybody wants, right? They're going to have opportunities to have symbiotic relationship with other organisms. Many microbes have evolved to have responses to starvation, environmental stress. So we saw in the first unit the formation of endospores. So they can actually have these morphological changes. Again, they can enter into that stationary phase of the growth curve. They can also use cellular components as nutrients. So we had talked how they have that slime layer or the capsule. So all these polysaccharides and proteins are on their surface, which is also referred to as the glycocalyx. So they can actually utilize that if needed. And they also have numerous genes and proteins that can help, right? So they have, and talking about proteins, we have different enzymes, so they have different ways that they can metabolize. We tend to think about metabolism being from a glucose standpoint, and we have to have glucose in order to metabolize and make ATP. Keep in mind that any metabolism chart is going to show you other aspects, right? So you can actually use nucleic acids, you can actually use amino acids, you can use lipids in order to enter into different points of cellular respiration to generate ATP. And we also want to keep in mind that it is sometimes a difficult distinction between viable and being dead and not culturable. And just to kind of emphasize this, you can see here on this figure from the textbook how when we think of something being viable and alive, it's going to be highly metabolically active. And then when we're thinking of something being dead, there's going to be these like cellular fragments. There's a lot in between that. And so it's not always this clear cut, right? Especially in microbiology when we're talking about a single cell, right? So we can have a cell that just basically takes down metabolism a notch, right? So it reduces its metabolic activity. We would still recognize that as being viable and alive, right? But we could decrease even additional metabolic activity. Its plasma membrane would still be intact. But it's not going to be producing as much RNA because it's not going to be making as much proteins, right? And then we can have it where the plasma membrane is still intact, but there's really no metabolic activity, right? It's kind of in a dormant stage. Again, it's still, we would recognize it as being alive, but it's just not metabolically active. And then going kind of deteriorating from that, then we could see changes where the plasma membrane is actually damaged. We can actually have the DNA degraded because again, it's slowing its metabolism down, and then eventually cell fragments and death. But again, there's lots of stages to get from viable and alive to dead, and depending upon the microbe and its environment, it becomes difficult to distinguish that. So at one meeting, it was a American Society for Microbiology meeting, the speaker was actually talking about this analogy of like your grandmother's hammer. And cells are very much like your grandmother's hammer. So there's that kind of just, at what point, so the idea is with the grandmother's hammer is that like, if the handle breaks on the hammer and you put a new handle on, is it still grandmother's handle? And it's like, well, no, it's still doing the same function. The head's still there. Well, okay, well, now what happens if the head of the hammer gets broken and you replace that? Because you replaced it over time, is it still your grandmother's hammer, or is it now a different hammer? And many of us would say, well, no, all the parts have been replaced, so it's no longer grandmother's hammer. The problem with cells, they're doing this replacement over time, and so it becomes hard to distinguish, is that the original cell? Because they turn their metabolism down so small. So if you're not in a nutrient-rich environment, you can have these cells that persist in this kind of, again, dormant state with very low metabolism going on. So are they still alive? But if we replace the nutrients, they're able to become metabolically active. So they were alive the whole time. But again, if we were to use certain markers, it would look like the cell is almost dead. Okay, so again, just to distinguish, it's not clear cut. All right, so then in the lab, when we're trying to mimic these environments, we want to make sure that we are growing, transporting, storing these microbes in conditions that, again, mimic their natural conditions, so that we're actually looking at how they would behave in their natural environment. And as I mentioned before, we're not always perfect at this, right? This is an area that groups are definitely looking at how to mimic those environments more closely to what they are getting in naturally, because we know if we change certain variables, it's going to change how that microbe grows. And so in lab, you're going to be looking at a lot of different types of culture media. And so each of those culture medias are going to have certain characteristics that have pros and cons to them, because some of them might mimic the natural environment really nicely, where others might be just a general purpose and allow for a lot of different microbes to grow, right? So we always have to be mindful about the environment we are picking for those microbes. Culture media can be solid. So agar, AGAR, can be added into the media to solidify it. So that's where you might have like a petri dish that you've added the media in, and it's going to be solid, kind of like a jello consistency, or it could be liquid. These are known as broths. So kind of like your chicken noodle soup broth, right? In both cases, whether it's solid or liquid, so whether it's an agar plate or whether it's a broth, that culture media is meant to provide the nutrients and things like pH that those microbes need in order to grow. Some of the media is going to have a known chemical composition. So if it's synthetic, it would be called defined. Other times, that chemical composition is what's known as complex. So defined is going to be like we take, like we ordered this from Sigma, we add 1 gram of it to half a liter, dissolve it, sterilize it, and use it. On the bottle, it will be defined of all the material that is in there, okay? Complex usually comes from like an outside source. So your blood sheep agar plates that we talked about with our case study, those would be known as complex because we're using material that there is certain variation between those. But like they're red blood cells, it's coming from blood, and so again, that would be complex. As I just mentioned with the physical nature, it could be liquid, solid, and we even have semi-solid, so they're not, it's lower concentrations of agar is usually used. We can have functional media, so we can have general purpose, so your nutrient agar or your nutrient broth, those are like a general purpose, so it allows lots of different types of organisms to grow in, and so a lot of your beginning inoculations would be done into general purpose. Eventually, what you will be using are selective or differential or enriched media. So these are special plates that the chemical composition is unique. There can be different chemical components in it that allow you to either select for certain organisms or differentiate certain organisms. So again, with the blood sheep agar plate, we saw that we could actually differentiate between different organisms that can undergo hemolysis. So they produce toxins that lyse the red blood cells. So we would be able to differentiate those different organisms. You will probably also use mannitol salt agar plates. So those are selective in that bacteria that like high salt concentration can grow on them. The plates also have a slightly acidic pH to them. So again, it starts to select what can grow on them. And you can also, it's also differential because if the bacteria produces acid, it produces a reaction because it has a pH indicator in it. And so we can actually see if it's Staphylococcus aureus that's growing on the plate because the plate differentiates between Staphylococcus aureus versus other species of staph. So again, this is going to be expanded upon in lab. So really interesting. There are different ways that we can measure microbial growth, the population, so we can do indirect measuring and direct measurings. And so for direct measurings, where we're doing direct counts, this is where we would view them underneath several ways that we can do this. One way is by viewing them underneath the microscope and using counting chambers. The counting chambers a lot of times are going to have grids. If you go into your textbook, they show you images of this, and I again wanted to kind of keep it a little bit simple. But you can, using that grid, you can count how many cells you have, and then by doing calculations, you can calculate the concentration. If you do this at different time points, you can, if there's an increase in the number of cells, that would indicate maybe we're in the exponential growth. If between two different time points the number of cells is the same, or the concentration of cells is the same, that would indicate you're in stationary. If there's a decrease, that would indicate that you're in death phase. One of the popular ones that are used are electronic counters, and so these actually have like a light that the cells will go through, and one cell at a time, and then using a laser, it can count how many cells pass through basically the eye, and it will give you a calculation of this is how many cells. Electronic counters are more expensive than counting chambers. So a counting chamber costs about like $200 and can be reused repeatedly. An electronic counter, like 10 years ago, cost about like $30,000 to $40,000, depending upon the company you got it from and service contracts and such. The electronic counters also have certain consumables, like you have to clean them, and so you need the cleaning solutions and such to flush out any cells that maybe are kind of stuck in the counting chamber in the device. So there's additional cost and upkeep for those. So many labs will use counting chambers. It does require that you have individuals that know how to use the counting chambers to do those. And then also a flow cytometry or FACS, cell sorting is also used because, like the electronic counters, you can count individual cells. And then if you had like a mixed culture, you could even divide the cells based on their size, which is determined in their scatter of the light. And a cell sorter, the flow cytometer, can actually separate them into different test tubes. So if you would want to separate them, you can also use different types of media to select for the different cells if you have a mixed culture too. All right, so hopefully this provides you an overview of some of the different factors that affect microbial growth, provides you with additional information upon the natural environment versus the lab environment, and lastly, a little glimpse of how you would count cells in the lab if you needed to, okay? Really important emphasis here is on the environmental factors that affect microbial growth, looking at pH, temperature, and oxygen. Those are really important components that you should know and you should expect are completely fair game when it comes to the exam. As always, if you have any questions, please don't hesitate to reach out to me.